moving on to modern technology and its effects on our DNA. So there's good and bad when it comes to this. So with modern technology and lifestyle changes, obviously with the more modern lifestyles that we live now, there are positives and negatives for our DNA. Some positives include very obviously improvements in technology, the ability to alter and treat certain genetic disorders and mistakes using gene therapy, other genetic engineering means, as well as since we have more knowledge about DNA and genetics, and we're figuring out our genomes, that has a variety that has an effect, positive effect on a variety of different scientific fields, not even limited to biology and biochemistry, but like evolutionary biology and even forensics and things like that. However, with our altered lifestyle over the last few centuries, with heavier use of things like tobacco and like preservatives in our food and chemicals that we commonly ingest, as well as possible exposure to radiation in certain parts of the world in certain situations, more exposure to smoke from like coal and burning of fossil fuels, that's also not healthy for you. We also have greatly increased our exposure to mutagens and carcinogens compared to people who were living obviously like 600 years ago, where they didn't have any of this and they were just living like essentially natural lives. They didn't have as much that like hurt their DNA, right? They didn't have the exposure to these mutagens and carcinogens that we have, right? At least in most cases. Obviously, certain mutagens and carcinogens have always existed. But now our lifestyles involve greater exposure to them. And this increases the risk of genetic disorders, most common of which is cancer, right? Cancer is a genetic disease through and through. Cancer occurs because you have a problem with your DNA, or there's something wrong with your cells or something like that, right? Most of the time, and a good majority of the time, cancer is caused by genetic mistakes. All right, but starting off with the good, CRISPR-Cas9. This is a very hot topic right now. I'm sure you've learned about this since you were probably in maybe 10 years old. When it was first coming out, you've heard about CRISPR-Cas9, at least if you're as old as me, probably, since you heard it since you were about 10 years old. The biggest scientific breakthrough in the past decades has been the discovery of the CRISPR-Cas9 system. Cas9 it's a natural bacteria that's natural protein that's found in bacteria, and bacteria have had this for a while, and it's used as a defense mechanism as viruses. When a certain target sequence, and remember how viruses work, viruses come into bacteria or I guess any cell, but in this case bacteria cells, they inject some of their DNA into the DNA of the bacteria, and then the bacteria will actually reproduce the virus's DNA for the virus, because it kind of hijacks its system, right? It kind of puts its own DNA in the bacterial plasmid. As a defense mechanism for this, certain bacteria have this Cas9 protein. When they see a certain target sequence, normally this is a viral DNA sequence, they bind to it and it activates the inactive Cas9 protein, which is a nuclease. So nucleases, remember, ACE means like usually cut or destroy. So nuclease means it cuts nucleotides, which is DNA, right? Nucleic acids. And once it's activated, the Cas9 protein, it then searches the bacterial plasmid, bacterial genome for the target sequence. Basically, it sees, okay, I have a virus. Okay, this is the DNA sequence of the virus. Now, let me search my own DNA just to make sure that this virus didn't infect me and put this DNA sequence in my genome. So it searches through it. If it finds that target sequence in the DNA in its own genome, it'll like a pair of scissors just cut on both ends and combine the two ends like light with a ligase or something like that, and it cuts out the DNA target sequence out of its own bacterial DNA. The target sequence, and obviously we can use this by kind of manipulating it and putting in any target sequence that we want to take from the bacteria and feeding it to this Cas9 protein. We can alter the Cas9, give it our own target sequence, and then it'll search through the bacterial genome and just cut it out for us and give us an isolated target DNA sequence that we want, right, to put into our other organism. The target sequence is then introduced into the human DNA, and using homologous DNA repair, the bacterial target sequence is introduced and incorporated into the human DNA, right? Let me see. So remember how homologous recombination works? You kind of can just put it in and use it kind of as like a template, and you can just incorporate it, and the DNA will just kind of put it in. Ideally, sometimes it doesn't exactly work like that. Sometimes you don't get homologous DNA repair, you get non-homologous repair, which is not ideal. But yeah, so we're trying to still figure some stuff out, but usually you want homologous DNA repair, which incorporates the bacterial DNA target sequence into our own DNA. And in this way, we can kind of essentially put in any target sequence we want into human DNA. Apart from this technology, which is the most popular and most talked about, we also use our new knowledge about DNA for pharmaceuticals. The number of therapeutic drugs for treatment of cancer has risen exponentially in the last decades and is continuing to advance much faster than almost any other medical field, because, again, cancer is almost entirely a genetic disease. How do you cure genetic disease? Well, you have to use genetic means. So there's a lot of therapeutics that are specifically targeting specific mutations and genes that are specific to certain types of cancers. For example, with our ability to kind of sequence and map out and use more genetics to determine cancers, we can see, okay, this is a kidney cancer, but what kind of kidney cancer? Is this squamous cell? Is this adenocarcinoma? What kind of cancer is this? Okay, now for kidney adenocarcinoma, what type of mutations does this one have compared to some other maybe kidney cancer or some other adenocarcinoma? What kind of mutations will it have? Well, it has these mutations. Well, let's see if we can use this drug which can treat this mutation or can deactivate or do something with this mutation, and let's see if this one works for this cancer. And then they do clinical trials and it works, and they're like, okay, well, we now have a pharmaceutical drug that specifically targets this DNA or this mutation that's found in this type of cancer, has a very good success rate, and it won't affect other cell types. It won't affect anything else. It'll only affect this specific mutation and this specific cell type, right? And that's ideally what you want, because obviously back many years ago, when you had chemotherapy, you would kill off a lot of unnecessary cells that are in your body, as well as unnecessary cells, which are the cancer cells. Additionally, DNA mapping and sequencing has become very useful for both the fields of forensics and evolutionary biology. With forensics, obviously, DNA sequencing is very important. You can find out what you can basically identify criminals. So if you're doing criminal research, you can identify criminals or something like that. It's also useful for other types of forensics, like, for example, even very basic that you probably see very often is like a paternal test or something like that, that uses modern DNA sequencing to determine, is the DNA a match or is it not a match with the child, to determine if the person is a father or not, is the father or not. In addition to that, it's very useful for evolutionary biology, being able to see and compare and sequence the DNAs of different groups of people and see where mutations, basically create a timeline of when did mutations occur, when did, for example, people from Europe differentiate from people in Asia. We can use algorithms and match up our DNAs to determine how did we change over time, when did these mutations occur, when did this migration of people occur, how did our evolutionary history go through, like what are the differences between groups of people, what are the differences between you and maybe your ancestor even 10, 20 generations ago, right? So that's very useful for evolutionary biology, and there's many, many more applications for this. Of course, with this also comes, especially with this CRISPR-Cas9 DNA editing, while it can be used for basically curing disease and helping, has the potential for medical uses, there's also the ethical component of how much should we change in our DNA. Is it ethical to, for example, use genetic engineering and this CRISPR-Cas9 system or any other gene therapy to, for example, change a child while it's in the womb, or do something that could alter like the DNA of a child before it's born, or even alter things that may not be diseases, but things that we want to change. For example, make a certain person taller, stronger, or something like that, right? It's not exactly a disease if you're shorter, but if, for example, somebody wants to be taller, how much, like, where do you go, right? So think about the moral implications of genetic engineering as well. That's one debated topic right now. Okay, now let's talk about the negatives of kind of like modern technology, modern lifestyles, mutagens and carcinogens. These are both very bad for you. The main difference between these two terms, they're fairly similar and they overlap a lot, but the main difference is mutagens are only substances that increase risk of mutations in your DNA once exposure to. For example, tobacco has a bunch of mutagens, like cigarettes and other cigars and other forms. They have a lot of mutagens in them. When you smoke them, they will increase the risk of mutation greatly within you. Meanwhile, carcinogens specifically increase the risk of cancer, right? Carcinogen cancer, whereas mutagens mutate. As I mentioned, cancer comes from DNA mutations, but that's not the only thing that can cause cancer. It is a major thing, but it's not the only thing. So not all carcinogens are mutagens. There are some carcinogens which are not mutagens, right? All mutagens are carcinogens, as I'm aware. All mutagens are carcinogens, but not all carcinogens are mutagens. There are some carcinogens that don't mutate your DNA, but rather they just change your cell division. They increase cell division. For example, substances which simply promote cell division but do not damage DNA. One common biological example of a non-mutagenic carcinogen is the virus HPV, right? HPV infection is the leading cause of cervical cancer in people. I think it's about 70% or something like that of people with cervical cancer had recent HPV infection, and it's one of the main reasons for that type of cancer. It does not lead to DNA damage or mutations, but rather it increases cell division of infected cells, of nearby cervical cells, which causes cancer, right? Because if they're dividing and growing a lot more, that naturally will increase errors in just natural errors in during gene replication, right? Right, because when you cell divide, you also have to replicate your DNA. Again, it's not 100% accurate replication, and we have repair pathways which greatly increase the accuracy, still not 100%. So if you divide a lot, it can cause mutations on its own naturally through errors, which will cause cancer. Many carcinogens also may have both a mutagenic and non-mutagenic component. For example, the smoke from cigarettes contains a wide variety of mutagens which can damage the DNA of various organs, including but not limited to lungs, throat. It can also affect, I believe, kidney, bladder, and a few others. It's not important, but just to let you know that it's not just lung cancer when you smoke. There's a bunch of types of cancer that happen when you smoke. But the smoke itself, when it passes through your throat and your lungs, it actually also damages, like physically damages the cells on your lungs and your throat. And when cells get damaged, just like if you were to get hit really hard, or something like that, or you were to burn yourself, you would have inflammation, right? During inflammation, there's increased division and production of cells. When you have a constant state of inflammation, for example, somebody who's a chronic smoker, is always smoking, always taking in, physically damaging their throat and their lungs, that area is pretty much constantly inflamed. During inflammation, a lot of cell division, a lot of production of cells, also not good. So you're both mutating the DNA as well as causing increasing cell division in that area. And it works basically doubly to cause cancer, right? So it actually it damages the DNA, which causes mutations, and then it also promotes the cells to replicate more, which can cause even more errors, and it can kind of increase the chance that a mutation will be passed on, right, to offspring of cells.